My name is Ravi Iyengar, and welcome to this course which will introduce you to systems biology. This course is part of a three-course series. In this course, I will introduce you to the basic concepts in systems biology, which is an emerging area of research and analysis in biological sciences. Systems biology combines both experimentation and a large amount of computation, and there will be two courses that will follow these. Avi Mayan and his colleagues will teach a course on the use of statistical models and network models in systems biology, and Eric Sobey and his colleagues will teach a course on dynamical modeling in systems biology. So what is systems biology? Biology itself is a very broad term in the context of this course. Biology is sort of encompasses from molecular to cellular to tissue and tissue/organ and physiological functions. So mainly in this course we will focus on mammalian cell biology as the basis for understanding systems biology. Systems biology is the study of how molecules interact and come together to give rise to subcellular machines that form functional units that are capable of the operations that are needed for physiological functions at the cell, tissue/organ levels, and so on. So the term systems biology started to be widely used in the early 2000s. Prior to this, this field was often called complex systems, and complex systems is an even vaguer term, and it encompasses studies not only in biology but also in physics and chemistry and social sciences like sociology and even economics, and it's very hard to sort of wrap one around what the field of complex systems really is. There are some methodologies, especially network analysis, that are common between all these fields, but nevertheless complex systems has sort of been a field that's sort of not been used, or it's too broad a term to be really meaningful. One way to think of how systems biology has come about is to sort of identify a few key papers, and I just selected few. There are many more one could sort of use to serve as examples of how systems biology got started in the past decade or so. In terms of experiments, the development of microarrays to measure the levels of thousands of mRNA simultaneously allowed us to see how many components in a cell change in response to a stimulus. The paper that I have cited was among the first papers that showed how the transcriptional program in human fibroblasts would respond to a stimulus. This sort of set the stage for large-scale data gathering. In terms of computation, there were several key papers also published in 1999. One paper that Upi Bala and I published showed how properties could emerge from interaction between biological signaling pathways, and how these properties, such as switching, could give rise to biological function. Another important property that is that emerges in systems is robustness, and a paper by Uri Alon and Nama Barkai and others in Stan Liebler's lab studied robustness in bacterial chemotaxis, and a combination of experiments and modeling, they were able to show how the system exhibit robust behavior in response to many perturbations. So one can ask the question, and people often do, isn't systems biology, especially in the context of mammalian systems, just physiology with a new name? And the answer is yes, up to a point. Physiology has always provided us with quantitative description of functions at the tissue and organ level, most often from a phenomenological perspective. These descriptions are very precise often and very useful and are an essential starting point actually for modern systems biology. So take for instance the filtration of urine in the kidney or the electromechanical activities of the heart. These have been described with great precision and in detail, but nevertheless these descriptions do not encompass the molecular and the genomic details that give rise to these observed physiological functions. So in classical physiology, molecular biology and biochemistry were not often fully considered in physiological descriptions. Systems biology starts to uses molecular biology and biochemistry of cellular components to understand how physiological functions at cell and tissue organ levels arise. Systems biology, as I told you, is a very sort of broad area of inquiry, and there are many branches to it, and it's worthwhile to sort of get some of these definitions and branches identified so that as we go through the course one can put these different areas of sort of research in perspective. And one of these most important ones is genomics. The term genome, which was sort of first used in the 1920s, refers to a set of chromosomes. And genomics considers genes in the context of whole genome, a little different from genetics, I guess, where one can say in genetics many times one can consider a gene by itself, what the characteristics of the gene are and how its function is affected by its intrinsic characteristics, rather than considering it in context of a chromosome or a whole genome. The difference is sort of overlapping and fungible. So anyway, genomics provides an understanding how genes are organized within the chromosome and the whole genome, and characteristics of genes such as single nucleotide polymorphisms, copy number variations, mutations, etc. There's also the field of epigenomics that deals with methylation of the DNA and how this regulates the expressions of genes. There is, of course, like I said, with microarray or the transcriptional profiling, and since changes in gene expression is a major way in which cells both maintain homeostasis and change state, patterns of mRNA expression is critical for our understanding of regulation. So one can consider genomics as one bookend of systems biology, because after all of the cellular components, the proteins or lipids, everything arrives from the characteristics specified by the genome. Lipids and other smaller molecules from the proteins that are made from that in turn synthesize or degrade them, and so genomics is one critical bookend of systems biology. The other end of systems biology would be the physiological functions, and one can think of that systems biology enables us to understand how the genomic characteristics, when they are expressed in terms of proteins, and how these proteins can then be further modified by environmental signals and other perturbations to give rise to dynamic physiological functions. So physiology and genomics can be considered two bookends of systems biology. Another term that we need to be cognizant of is the term multi-scale. In the context of this course, multi-scale refers to the scales of organizations. Molecular components, for instance, molecular components to subcellular machines, transcriptional machinery, self-motility machinery, and so on. These subcellular machines in turn go to form cells, cells to tissues and organs, and interacting organs to whole organisms. So the increasing levels of organization give rise to new properties and capabilities, and of course systems biology involves the study of how these new properties and capabilities arise, and so understanding this multi-scale organization is sort of an intrinsic part of the study of systems biology. Sometimes multi-scale, in the context in which we work, also refers to different time scales, such as milliseconds to seconds, to minutes to days, and this reflects sort of, say, the activity of channels that open and close at the level of milliseconds, but the activities of the channels may give rise to changes in gene expression, or the epigenomic status of the chromosomes give rise to changes that are expressed at the level of days or even weeks and months. A good way to think, or a very broad classification of how systems biology approaches can be bent, is to think of top-down and bottom-up approaches. Top-down approaches start from descriptions of the system as a whole, understanding systems' characteristics and capabilities. Typically, top-down models provide a big and sometimes comprehensive picture of the system. However, relationships in top-down models often are defined by correlations, and causal inferences is not possible in the scale at which these large models are described. So this is sort of a limitation of these top-down models currently. In contrast, bottom-up models start with cellular components such as genes, proteins, or protein products such as lipids and sugars, and develop an understanding how functional systems such as subcellular machines are assembled, controlled, and operated. Bottom-up approaches have been what has been most common in molecular biology, biochemistry, cell biology over the last 50 years. This approach is sometimes called reductionist, starting reducing the system to one key component and then starting from that component of interest and seeing how the rest of the system develops. So the bottom-up approaches are good in that they provide mechanistic understanding, tell us how things work. So this is sort of a very important aspect, positive aspect of bottom-up approaches. But as the systems gets larger, one can often get lost in the details, and one cannot really decipher how a small detail in relationship to one or a few components relates to systems behavior as a whole. It's sort of a case of one can't see the forest by just looking at the leaves, and although this sounds like a cliche, this really is true. So in reality, one needs a combination of both bottom-up and top-down models to give a useful perspective in systems biology. And as we do our course, as we go through the course, you will see how this becomes apparent. This sort of concludes this first half of this course, and in the next part I will discuss some more of these terms and how large data sets are organized and where we want to go. Thank you.